Thanks for the introduction. Hi, I'm Paige. I'm a PhD candidate in Dr. Hao Chen's lab, and I study social isolation in adolescence and how that plays a role in oxycodone use in adulthood. And so I think we're all aware, can all agree that there's many factors that influence whether or not somebody gets a complex trait such as, am I talking too close to it? Okay.
Let's continue and see if it's a problem.
All right. So there's many factors that influence whether or not somebody gets a complex trait, such as addiction or substance use disorder. And just to name a few of them, there's education, resources, finances, other social determinants of health. And I think we all can also agree that there's, sorry.
If somebody in this room might have on Zoom, you may have your mic on. That may be the source of the problem. Or just back up. You're fine.
All right, everybody can, if we're good. All right. All right. All right, so I think we all could agree that a lack of some of those resources can be chronic stress or a chronic stressor. And one type of chronic stress is social stress. And we also know that gene expression can be impacted by chronic stress. And so I specifically look at the very specific social stressor of social isolation. And I also look at the critical developmental window of adolescence. And I do this by trying to model this in rats. And this is my experimental timeline. So after the rats are weaned, they're put into two groups, either group housed or isolated. And the group housed are in pairs. There's 2 per cage, and then isolated are just as they sound. They're alone in the cage, and they stay that way for 6 weeks. They're not interrupted. There's no other recordings or behavior taken. And then after that 6 weeks is up, the rats that were isolated are paired together, and they start a 3-day familiarization period. And that is essentially just so they can get used to their environment and their new cage mate. And the rats that are group housed, they stay the same. And then they start self-administration. And so I also take behavior data, and this is when I take baseline behavior data right before the rats are paired. And I do that because I'm going to compare the baseline to after a self-administration session. So we call that post-drug, and then also during a withdrawal period. And so I take things like tail immersion assay, I take blood plasma to look at Cort levels, I take elevated plasmase, and I also take social interaction behavior using a camera and machine learning. And so I also do this in 2 strains to make sure that our data is not strain specific. And we use inbred strains because we are going to look at gene expression. I use the WKYs because they're supposedly a model for anxiety and depression. And they have supposedly a genetic predisposition for anxiety and depression. And now the DSS, certainly not in our lab, but in other labs, they're used as a model for hypertension or salt sensitivity. But for us, what's important is that they don't have a background of having anxiety or depression. And so essentially they're used as our controls to the WKYs. And so before I go into my self-administration data, I wanted to go a little bit into my baseline behavior data for elevated plasmase, because I do have some statistical significance even before the rats start taking drugs. And for those that don't know what the elevated plasmase is, it's this picture up on the left, and it's a platform that's it's up to my waist, probably a normal person's height, knees. And so there's 4 arms. There's 2 arms that are closed, they have walls on the side, and then 2 that are open where the rats can fall off. And so it's used as a measure of anxiety or anxiety-like behavior in rats, because the rats that are more anxious are going to spend more time in the closed arms and not take that risk to go to the open arms. And then the rats that have less anxiety are going to spend more time in the open arms. And so for my data, the WKY females, do I have a mouse? I don't know. Yeah. All right. So that plot, so the rats that were group housed in adolescence, they show more anxiety-like behavior. In other words, they spend more time in the closed arms than the rats that were isolated, which is the opposite of what most people would expect. I think most people think that rats that are isolated would have more anxiety, but we do see a general trend in at least 2 other plots as well, except for the DSS males. And then this is our oxycodone self-administration boxes. We call them PurePub, and we build them in-house. And that's because we wanted to develop a model for rats to be able to take drugs socially. And so essentially, there's other papers that have tried to do something similar where they put maybe a wall in between them or a glass in between them, or they'll have only one rat hooked up to drug. But we wanted to be able to record 2 rats simultaneously while both being able to get drug and then record which one is taking the drug and when and how much. And so the way we do that is we implant an RFID into the top of their head, and then there's an RFID scanner around each spout. And so when the rat pokes its head in, it'll scan that RFID, and that's how we know which rat is which. And so just about everything else in the operant chamber is similar to other operant chambers that are used. You have your active and inactive spout, you have the optional water spout, and the pump, we also build that in-house. It's run by a motor, and then everything is controlled by this Raspberry Pi here. Everything is recorded in the Raspberry Pi and controlled using that. And then the other thing that I don't have in this picture that is an option is we can move that Q light off to the side, and we've put a camera above it, and then the top has clear glass you can see through, and then we can also record social behavior and social interaction while the rats are taking drug. And so I know for a lot of people this can be kind of confusing if you aren't seeing it. So I put a video in here, and I'm not sure if it'll play, but hopefully it will. Okay, so yeah, when the rat puts its head in there, it's, if you can see the Raspberry Pi on the side, there we go. So it'll count how many active, inactive, and then the amount of rewards. And then it has the RFIDs, if you can see them here as well. So which rat that was. That blue light was the cue to know it had a reward, and it's on a fixed ratio of 5, which means that it takes 5 licks to get a reward and a timeout set of 10 seconds. And the other thing that this box does is it also can distinguish between actual licking of the spout and then of nose pokes, because a lot of rats are curious and they'll just poke their nose through, but it can distinguish the difference and then count how many nose pokes and count how many licks there were. And so this is my actual oxycodone self-administration data. So the way we do this protocol is we start rats on a 1 hour, hold on, let me point to it. So if we use this as an example, we start them on a 1 hour consecutive everyday session where essentially it's just so the rats can get used to the boxes and learn how to take drugs and poke their nose through and lick on the spout. And we start them on a very low dose with that. And then we move up to 4 hours, and we also do every other day. And that's so we can create a withdrawal period. And so essentially a rat will have a session, and then behavior can be taken after that session, and then we can have an off day or a withdrawal day, and withdrawal data can be collected on that off day. And then we keep on moving with the 4 hours, but we increase dose up to 0.1. That's the max that we use. And that's really essentially because once I went higher than that, I think the rats maybe thought the oxycodone was too bitter at that point because they started taking less. So really that's the dose where I saw group differences in. And if you haven't already been able to tell, the between both strains and sexes, the rats that were isolated in adolescence do take more oxycodone in adulthood than the rats that were group housed in adolescence. And there's no differences between the groups at this point. The only difference is basically just their adolescent period, whether or not they were isolated or group housed. At this point, they are all taking drugs socially. And so this is my tail immersion assay that I wanted to show. So essentially the way this works and the reason we use it is we want to make sure that the oxycodone is working and that it has an effect. And the way we do this is by putting the rat's tail in hot water and then seeing how long the rat's tail can stay in hot water. And we call that latency. We record how many seconds. And we do this at the baseline that I showed earlier, and then also during the withdrawal, and then also right after a drug self-administration session, which is called post-drug. And so because oxycodone is a painkiller, you'd probably assume that after taking the drug, the rat can keep its tail in hot water longer. And that's exactly what we wanted to look at and what we saw. So the more the rats took, the more oxycodone the rats took, the higher the latency, or essentially the longer they can keep their tail in hot water. And all of them were significant except for the WKY males. And so the reason for that is essentially we noticed that the females tend to take more oxycodone than the males, and then also the DSS take more oxycodone than the WKY. And so I think for the WKY males, they unfortunately did not take enough oxycodone to have an effect, to have that analgesic effect. And so after the self-administrations are done, we usually do about 17 sessions. And so on their last session or their 18th session, 1 hour in, we take their brains and then we dissect out brain regions. And we dissect out brain regions that have some sort of role in drug use or drug addiction or social behavior or social isolation or all of the above. And these are the ones that we went with. And so we wanted to look at CFOS relative expression differences between the two groups, or essentially neural activity between the two groups. And we did see that so the nucleus accumbens shell and the infralimbic cortex, those do have statistically significant differences between the groups. The rats that were isolated in adolescence do have more neural activity or more relative CFOS expression between the two groups. And so right now where we're at is we are sending those samples off for RNA sequencing because we're going to look at gene expression. And unfortunately, I don't have that data now. And I'm also still collecting these CFOS data as well. So can't really show any of that, even though I know it is a genetics conference. But I just want to thank my lab and my awesome and supportive mentor and my committee members and ask if there's any questions.